Hello, my name is Andres Maldonado, and today I'm going to present the RNA transcription in Eukarya. The cycle of eukaryotic transcription from initiation to elongation and termination is regulated as multiple steps. The mechanism of transcription regulation is much more complex in Eukarya than in prokaryote. Although, because of the presence of nucleus, the RNA processing and the presence of histones and the highly condensed regions of chromosomes. Just like the cell cycle, the transcription cycle can be divided into a number of distinct steps. In order to RNA-P2 to transcribe a gene, it needs to be recruited to the promoter, assemble with the general transcription factors, and then initiate the transcription complex assembly, open complex formation, and initiation. Because these early events have proven to be a main target for the mechanisms and factors that regulate transcription, they had been studied in greatest detail. Regulation and transcription obviously occurs into the context of chromatin, adding yet another layer of processes that is prone to regulation. In order to cope with the fact that DNA template is embedded in a chromatin RNA-P2, transcription does also entail recruitment of chromatin remodeling complexes, such as the ATP-dependent chromatin remodeling machines and histones modifying enzymes to facilitate the process. The first phase of gene expression is the production of an mRNA copy of the gene. As all other RNA, and messenger RNA is formed on a DNA template by a process of transcription. Transcription is initiated when RNA polymerase binds to a specific DNA sequence, called the promoter, located at the 5' end of the DNA, which contains the start site of the RNA synthesis and signals this process to begin. After binding to the promoter, the RNA polymerase opens up an adjacent area of double helix to expose the nucleotides on a small stretch of DNA on each strand. One of the two exposed DNA strands serve as a template for complementary base pairing with RNA nucleotide. Therefore, guanine, cytosine, thymine, and adenine in the DNA would signal the addition of cytosine, guanine, adenine, and uracil, respectively, to the RNA. The RNA polymerase then moves stepwise along the DNA helix, exposing the next region of DNA for complementary base pairing, until the polymerase encounters another area of special sequences in DNA, the stop terminal signal, where polymerase disengages from the DNA and releases the newly assembled single-stranded RNA chain and both of the DNA templates. The RNA chain is the complementary to the DNA from which it was copied is called the primary RNA transcript. The primary RNA transcript is approximately 7 to 10,000 nucleotides long because only a selected portion of DNA is used to produce the RNA molecule. Primary RNA transcripts vary greatly in size because of the presence of long non-coding intron sequences. This is in contrast with mature, more uniform, small-size RNA sequences that are needed for encoding proteins. The primary RNA transcript into this cap by addition of methylhalate guanine nucleotide to its 5th end. The 5th cap always plays an important role in protecting growing RNA transcript from the degradation and later in the initiation of protein synthesis. The 3rd end of primary RNA transcript is cleaved at the specific site, and poly-A tail is added by poly-A polymerase. The poly-A tail facilitates the export of mature messenger RNA from the nucleus influences the stability from the messenger RNAs into the cytoplasms and serve as a recognition signal from the ribosome, which is required for translation of messenger RNA. From messenger RNA to move out of the nucleus, primary modified RNA transcripts undergo one more RNA splicing events. The non-coding sequences are removed by ribonucleoprotein complex, and the coding sequences on either side of the introns are joined together. These events result in a small, relatively stable, mature messenger RNA, which accounts of approximately 3% of the quantity of cellular RNA. The poly-A tail facilitates the export of mature messenger RNA from the nucleus, influences the stability of some messenger RNAs in the cytoplasm, and serves as a recognition signal for the ribosome, which is required for translation of messenger RNA. From M messenger RNA to move out of the nucleus, primary modified RNA transcripts undergo one or more RNA simplicifying events to the not coding sequences are removed by ribonucleoprotein complex, and the coding sequences on either side of the introns are joined together. These events result in a small, relatively stable, mature messenger RNA, which accounts of approximately 3% of quantity of cellular RNA.